All right, so here we are in lab 4. I'm kind of going to repeat myself for those of you who are here. I just had to stop the recording, but your lab 3 quiz will be due this Sunday, and that lab 3 quiz will have pictures identifying organelles in the cell, as well as pictures of identifying different types of tissue. And I use the same pictures from the lab guide to identify those tissue types. It'll be completely multiple choice. And then this week we have our cell chemistry, diffusion, and osmosis lab due on Sunday, September 19th. And just a note on this, you'll need to download the PowerPoint, and you'll need to view the PowerPoint in presenter view to view some of the animations or videos within that PowerPoint. So I'm going to go ahead and just show you an example of what I mean by that. So here's the PowerPoint that you need to download. And I think the first video is on slide 15. And on slide 15, when you hover over this picture, you should have a option to play a video, which is just showing, in this case, heating up the vials that are in warm water. So just keep that in mind. If it asks you to watch the short video clip, you'll need to download the PowerPoint to play that video. So that should work for you guys. I'll go back to the beginning now and share the worksheet first of all. So this is actually a really fun and interesting lab to do in person, and the head faculty leader who made all your online labs actually took you through what we would be doing in person, and that's what she takes you through. That's why the PowerPoint guide, it's 46 slides, but it takes you through step by step what you would be doing in person. And this is a really interesting lab to look at the different tests that you can perform to detect the presence of specific chemicals in a solution. So you will be performing different tests using different indicators. You'll explain the mechanisms by which the tests are used to specifically determine the presence for sugar, starch, and protein. So you'll be performing different tests to determine if a solution has sugar in it, if it has starch in it, and if it has protein in it. And then you'll apply this knowledge to different structures. Good, Valerie, I'm glad you can hear me. Normally it's on my end, so always let me know if you can't hear me.
Yeah, I can hear you now. I just didn't catch what you said in the beginning. Did you say anything about the video link?
Yes, so what I mentioned in the video link, that in the PowerPoint, you just have to download the PowerPoint as shown here. And then when you hover over a picture in the PowerPoint, it'll give you an option to play a video.
Okay, I'm gonna go. I have another screen right here. I'm gonna try to get that.
Yeah,
Sorry, I didn't mean to interrupt you. Yeah, because I downloaded it like you had asked on the email, and then it didn't. For some reason, it still didn't come up. So let me try it again. You said all I have to do is just download it, right? And pull it up in the PowerPoint.
Yeah, you just need to open it as a PowerPoint file on your computer, and then it should work. But let me know about that, Valerie. But yeah, it should work. The nice thing is the videos aren't super. You can still do the lab without seeing the videos. The videos are just kind of showing you a simple step. But yeah, let me know if that works.
Okay. Yeah, I was even just gonna go on YouTube, and there's other videos that have the same. Pretty sure that have the same demonstration.
Yeah.
Okay.
So yeah, so that's really important. If you want to watch the videos, just download the PowerPoint. But this lab takes us through looking at the 4 basic classes of molecules, which are carbohydrates, proteins, lipids, and nucleic acids. And this lab uses what we call indicator chemicals or reagents to detect the presence of different classes of these molecules in water solution. So for example, the lab guide will take you through, it'll have like a little vial with a solution in it, and it might have sugar, starch, or protein in it. And then you guys will add different chemicals, whether that's IKI, Buret's reagent. And if you see a color change or some sort of change that the lab guide will describe for you, that means if there's a positive reaction, it means that that particular molecule is present in the solution. And if there's a negative result, meaning no color change or whatever else is an indicator, that means that there is not the presence of that molecule in the solution. We use control reactions to always be performed as a comparison. And usually the controlled reaction just has nothing in it, distilled water. And as it takes you through these activities, it'll take you exactly through. The lab guide will show you pictures of what will be done to detect the presence, for example, of starch in a solution. So the first activity detects for the presence of starch in the solution. Starch is carbohydrates, so we'll be detecting for sugars, and it gives you the materials, and then it'll show you pictures of using those materials. I'm going to kind of go through and then take you to table 1. So table 1, you'll need to put the reagent color, which will always be the same. That'll be the reagent color, which is IKI. And then you'll show the color of the sample after you add the sample. And then you'll just say yes or no, a color change. And then the conclusion means you'll put positive or negative if a starch is detected. So what I mean by that, let's take you through a little bit of activity 1. So here's activity 1 and all the materials shown. So activity 1, you're testing for the presence of starch in a solution, and this vial has glucose in it, 1% starch, distilled water, and then IKI is the chemical that will detect for starch. So you'll first label your test tubes. One will be distilled water, that'll be the control because there's nothing in it. One will have sugar in it, and one will have starch in it. So you'll first add, I think, note that 5 drops of DI water are being added to the DI tube. So I think what happened first is we added the reagent, which is this kind of brown color shown here. So the reagent is IKI. That means this is what's been added to your test tubes first is IKI, the reagent. This is the chemical that will be able to detect for the presence of starch. So when it asks for the color, the reagent color on the column in table 2, in the table on page 2, you'll just want to put the reagent color is kind of a, I don't know what you would call it, a golden honey color. So you'll put the reagent color, is golden, yellowish. Then we add all of our solutions. We're adding distilled water, we're adding sugar, we're adding starch, and then we will watch this short video clip to see what happens. And you'll see what the color of the solution does. When starch is added, you can see that the color of the solution changes from yellow to kind of a black or a very, very dark blue. And that means that this was positive for starch. And that makes sense because we added starch, so the IKI reagent will detect starch in a solution. So here are the results. Again, distilled water, we just have our IKI reagent with distilled water, and there's no reaction. It stayed the same yellowish honey color. So that means that there's no starch detected. But in the starch beaker, the one way on the right side, you can see that it changed color so we had IKI, the chemical reagent, and then when we added starch in, the IKI, the reagent, was able to detect that starch, and it changed color to dark black. So when it asks you for the color after the addition of the molecule, you can say that in the starch column, it changed color to black, and that gives you a positive reaction, meaning that IKI was able to detect for starch in the solution. The same thing occurs detecting for the presence of sugar. So we use the same kind of 3 solutions. Distilled water is our control. We'll test for starch again and then test for glucose. But now the difference is that we're using a different reagent because each reagent tests for a different substance. So for example, the IKI, which is what we just tested, that will detect starch in solutions. The Benedict's reagent, which is in blue here, let's see what Benedict's reagent tests for. So we set it up the same way. We put our Benedict's reagent in first, and at the beginning, you'll want to note that it's blue. The Benedict's reagent is blue. We'll add distilled water to our first test tube because that's our control. So we shouldn't see any color change when we add distilled water because there shouldn't be anything in distilled water. We'll add sugar, we'll add starch, and then we'll test for the presence of sugar in the solution. And in order for Benedict's reagent or Benedict's solution to work is it needs to be heated up. So this shows the idea that we've heated up these 3 test tubes in a beaker of water that's boiling. And you can see here that one of the beakers has changed color. In the presence of heat, Benedict's solution will detect for the presence of sugar. And that's what this video shows, that when we heat up these vials with a solution, one of them will change color because the Benedict's solution needs to be heated up in order to work. And that's what this shows. It shows that the glucose or sugar test tube changed color in the presence of heat. So the Benedict's reagent tested for sugar, and we know that the Benedict's reagent tested for sugar because after heating it up, the sugar test tube changed color. And again, it'll ask you for the color that it changed, and you could say reddish, dark orange. So that would be a positive solution or positive presence of sugar due to the Benedict's reagent. Any questions so far?
Yeah, I have a question. So pretty much, so for like the reagent color and the Benedict, that on that column right there, it'll all be the same because it's all using the same solution, right?
Yeah, so whenever it asks for the reagent color at the first column, it'll always be the same because that's what you're starting with. So for example, this is activity 1, the reagent color of IKI, that was like yellowish or honey. And then after we added the sample, one of them changed color. It was the starch, and it changed to like black. So that gave the positive indication that there was starch there because the reagent changed color. Activity 2, the table will be set up the same way, but we're using Benedict's. So the reagent color is blue. So you'll put blue, 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 or light blue if you want to be really detailed. Then you'll do the color after the addition of a sample and after you heat it up. So water stayed blue, amylose, starch stayed blue, but glucose after heating, it changed color to the kind of that dark, the reddish, dark orange color. So that was a positive indication for sugar. And then we'll do one more test, but now we're using protein and we're going to use Byerowitz reagents. So now we're going to use a different chemical to test for the presence of protein in a solution.
So excuse me, so the reagent that the solution, that's pretty much what'll detect for each single molecule that's contained.
So good question. Different reagents detect for different molecules. So Benedict's detects for sugar. So that's why the vial with sugar in it changed color. And IKI, that reagent detects for starch. So that's why the vial with the starch in it changed color.
Okay, I'm getting it now.
Yep, no, so I would say this is a little confusing. So even like even watching this video again might help. But yeah, it is a little confusing at first. And we're going to do it a third time. So this time the reagent, which again is just another word for a chemical that detects the presence of a protein or whatever in a solution, we're going to use what we call Byerowitz reagent, which detects for protein in a solution. And we're going to put 2 solutions. Distilled water is our control, remember, because that doesn't have anything in it, so it will never change color. And in the other kind of test tube, we're putting our protein in it. So we're just kind of, again, this activity is just showing you that different chemicals detect different molecules in a solution. So you'll notice at the beginning, the Byerowitz reagent is also like a light blue color. When we add distilled water to it, it'll stay blue. But when we add protein, let's see what happens and how the color changes when we add protein to it. So we add the gelatin, which is the protein, to it. We'll watch this short video clip to see what happens. So you'll notice we're adding protein. And you can see here that the Byerowitz reagent detected that protein because it changed color. You can see it changed. Let's watch it one more time. When the protein was added, it changed from kind of a bluish color to a purplish color. So that color change, again, indicates a positive presence of that molecule in the solution. So this just means that the Byerowitz reagent detected the protein because it changed color. So again, here's looking at the control with the Byerowitz reagent was light blue, and then the protein, the Byerowitz reagent was able to detect that protein because when we combined the protein with the Byerowitz reagent, it changed color to light purple. Right?
So Ms. Bemmer, I noticed that you are on a that's the PowerPoint that you're showing, you're demonstrating this on us. And when I downloaded it into my from the Canvas, it downloads into a PDF. So it's not download. So when I go like to look at the videos, I don't have that option how you do in for the PowerPoint. So pretty
Let
much.
me just double check. Yeah, good question. That could be my fault. So I have two. Let me just double check that.
I don't know if other students have been able to see the video.
No, I think you're right. So no, Valerie, this is why I only uploaded the PowerPoint to one section. When we're done here, I'll uploaded the PowerPoint to your section too.
Yeah, because I'm downloading it, but it's downloading as a PDF. It's not downloading as a PowerPoint.
Yeah, so that is my fault. So after yep. So, and that's my fault. So I had two sections of you guys and I did not upload the PowerPoint. So that's why you're having so much trouble, Valerie. It's all my fault.
That's okay. I'm always calling you out, huh?
No, thank you so much. So right after this, Valerie, let me get through the recording and I'll upload the PowerPoint right after this, okay?
All right, cool. Thank you.
Oh, sorry about that. Yep.
That's okay.
Like I told you guys, like 99% of the time if something goes wrong, it's my fault. So yeah, I will upload this PowerPoint. So let's try to get through. This takes you then through the activity 4, which tests for the presence of lipids. You'll use brown paper squares. You'll put a drop of vegetable oil, which will be the lipid, compare it with water and kind of what you see when you look at the brown paper. Diffusion through a semi-permeable membrane. This is a fun activity too. What we do in this activity is we use what we call dialysis tubing, which means that some things are able to get through and other things are not. And we're going to use our IKI reagent to try to detect for the presence of starch. Some of this I might have you guys work on through your own because it takes you through this setup. We soak the dialysis tubing, we seal it off. We will add 1 solution of glucose and starch into the bag. So we're putting 2 molecules into this bag, glucose and starch, and we're going to try to see if glucose or starch will leave the dialysis tubing, if it will leave the bag. And we're able to do this. We'll measure the weight of that bag because we want to measure to see if anything leaves. So you'll have to indicate the mass of it. We are going to place the bag in a beaker containing a solution of distilled water and IKI. So just taking a step back here, we've kind of created this bag with a semi-permeable membrane, which means that some molecules will be able to get through the bag and leak out into the surrounding beaker. Remember, inside the bag, we put starch and glucose. And outside the bag in this solution, we have the IKI reagent. Remember that your IKI reagent will detect for the presence of starch. So if we see any color change, either inside the bag or outside the bag, that will tell us if the starch stayed inside or leaked out into the solution if we see a color change, because the IKI will change color when it detects starch. We will measure another mass. The second bag will be filled with protein because we're going to also try to detect protein. We're placing this in just distilled water to see if protein will move in or out of the bag. After 1 hour, we noticed that the inside of the bag changed color. It changed that dark black color. And remember that dark black color occurs when the IKI detects starch. So what this tells you is that IKI was able to get into the bag and detect that starch. The starch, however, was not able to get out of the bag. It was too big of a molecule to get through the holes in the bag's walls. So what this is showing you, just that IKI was able to get into the bag to detect the presence of starch, but the starch was not able to get out of the bag. So you will kind of weigh both bags again. Here's bag 2 at the end of 1 hour. So you'll just notice maybe any change in difference of weight to notice if any protein was able to leak out or into the bag, because a difference in weight will show you if it has more water in it or less water, or more protein or less protein. And this kind of takes you through this diffusion experiment. You'll test beaker 1 solution for the presence of sugar. So you'll use the results from part 1 of this lab to determine if the image on the right shows a positive or negative result. So we're trying to detect the presence of sugar. You'll test for the presence of protein, to look for any color change. And then we'll look at the effect of solute concentration on osmosis. This one's a little bit easier. This is the last activity you'll do. Basically, what you're doing is you're taking plant cells, the Elodia, and you're placing some plant cells in a salt solution. So you'll look at Elodia. These are plant cells before being placed into a salt solution. And these are plant cells, the Elodia, after being placed into a sodium chloride solution. And you'll notice right away that the plant cells in the sodium chloride solution kind of get bigger. They kind of take in water. What this is kind of referring to is it's showing how osmosis will enter in to an area where there's more solute concentration, and it'll just kind of increase the size of those plant cells. So I'm going to kind of stop it here, but I'll answer any questions. So I'll stop the recording. Like usual, for those of you who are watching the recording, if you guys have questions on this, send me a message. Come to my office hours Thursdays at 11:30, and good luck.